Hello everybody, this is Dr. Vishal Trived from Department of Biosciences and Bioengineering, IIT Guwahati. And in the previous lecture what we have discussed about the applications of biotechnology in the agriculture science. In that lecture we have discussed about how the genetic engineering as well as the biotechnology is allowing to generate the different types of plant species which are resistant for the disease as well as resistance for the abiotic stress, and all these advantages have improved the crop production as well as they have increased the yield from the same plants. Now, in today's lecture we are going to discuss about the role of biotechnology in the animal sciences. So, animal science within the animal sciences, what we are going to discuss about the role of biotechnology in the animal breeding, and within the animal breeding the biotechnology has greatly facilitated the animal breeding and improving their species with additional traits uh, by discovering as well as validating the two processes. One is called artificial inseminations, the other one is called as the embryo transfer, and both of these processes are having its own significance, its own importance in the whole process of animal breeding as well as improving the particular species with the desirable traits. So, in the artificial inseminations, the overall process involves the introduction of male sperms into the reproductive tracts of the female animal artificially. The availability of superior breed animal is due to the artificial inseminations. So, there are multiple steps which are involved in artificial inseminations. In an artificial insemination procedure, the semen is collected from the superior breed male in a test tube. The quality of the sperm, such as the motility, the number of sperms in the semen is checked through microscopic observations, and then it is mixed with the extenders such as milk, yolk, glycerine as well as the antibiotics, and then stored in a liquid nitrogen for future use. Now, before you go for the artificial inseminations, it is important to study the estrous cycle of the female to know the exact time of ovulation, because it is important that you should know the estrous cycle of the receiving female counterpart, so that when you do the artificial inseminations, the sperm what you are going to inject artificially are going to go and do the fertilizations, and that is how you are going to have the better traits with the help of this procedure. The semen is injected into the female reproductive tract to facilitate the process of fertilization, and because this is a very difficult task, a skilled technician or the doctor is required to perform the most of the steps of artificial inseminations. There are several advantages of artificial inseminations compared to the natural breeding. So, one of the major advantage of the artificial insemination compared to natural breeding is that you could be able to introduce the desirable traits whichever you like to. So, in that case what you have to do is the male of a hybrid, which means the breed which is desirable, which is required for different types of purposes. For example, if you have looking for the breed which should be very powerful in terms of using it for the agriculture purposes. So, the breed what is going to be used for artificial inseminations is known as the Cyrus, and it is very costly in comparison to the semen from them. So, if you would like to buy the same breed from the same breed after it is going to be very costly, compared to that you can buy the a vial of semen from the same breed, and that was going to be cheaper. So, if you use the semen, you could be able to perform the artificial inseminations, and that is how actually you are going to get the similar breeds even in this procedure as well. Through a natural breeding process, many diseases can be transmitted to the female through mating. these possibilities are much reduced in a AI procedures. So, when people go for the natural breeding, there are so many sexually transmitted disease which are going to go from the male to female during the fertilization steps. So, that kind of possibility can be reduced very significantly, because you can easily check the semen as well as the quality of sperm in a microscopic observation, as well as we can perform many of the biochemical as well as the microbiological observation as well, to see if the semen is actually contaminated with any kind of the infectious organism or not. The hybrid animals imported from the other country need to go through a quarantine process to ensure no spreading of disease. So, what happen is, when even if you would like have a money and you would like to buy the hybrid from the neighboring countries, when these breeds comes to the our country, they have to go for a quarantine process. Quarantine is a process in which the animal is going to be kept in isolations, and it is also going to be under the observations for the appearance of any kind of diseases what the animal is going to have. So, in the during this incubation procedures, the first of all, the quarantine process is very costly, because the animal has to be monitored throughout this incubation periods, and it actually so because of that and it is also a time consuming process. So, this process is very costly and time consuming. So, you have the three advantages. First, you do not have to go with the hybrid animals, you can easily take up their semen and go for the artificial inseminations. So, with the help of expert technicians, you could be able to get the same breed, but without going through the high cost of that particular breed. The number 2 is, because you are using the artificial inseminations, you could be able to control many of the sexually transmitted disease from the male to the female and so on. And the third is that you do not have to keep these animals under the quarantine process. So, you could be able to save the time as well as the money by following the artificial inseminations. But there are disadvantages of artificial inseminations. What are these advantages? See, we are living in a country, so we are having a inbred animal species, and the major advantage of these inbred animal species is that they are very resistant for the diseases which are occurring in our environment. For example, if you are buying if you are going with any kind of breed which is coming from Australia or America or Europe, they have never been exposed to these diseases. So, they are good in terms of the food milk productions, they may be good in terms of their power, they may be good in many other traits, but they may not be very good in terms of sustaining the infectious organism what they are going to get in this environment. So, because these animals are never been kept in a tropical country like India, they will never been acclimatized for these diseases. So, because of that, the species what we are having, which actually may not be good in terms of the economical point of view, but they are very good in terms of the disease conditions or withstanding the environmental conditions. For example, our animals could be good in terms of withstanding the very high temperatures, very low temperatures, and as well as very high quality of humidity, and all these conditions are actually going to make the animals very sick in due course. So, even if you go with the artificial inseminations, you will keep going to get the hybrids which are going to give you the good milk productions, they might be economically very happy, but these animals may actually going to reduce the number of the natural breeders present in the vicinity, and as a result, if you reduce our own animals in this course, you are actually going to lose the traits which are available in these animals, and these traits are important because some of these traits might be very important in terms of providing the disease resistance. So, this is what you are going to lose the natural traits, like the natural traits to combat the environmental conditions and as well as the natural traits to take care of the disease. The second procedure is the embryo transfer. So, in the embryo transfer what you do is you actually take a superior breed female normally known as dam and can be able to give the more offspring in a year by a process of embryo transfer. So, what will happen is you are actually having a superior breed female, but a female has a limited number of babies which it can actually produce in a single year. But if you want to increase the productivity, what you can do is you can generate the embryos in a superior female, and then what you can do is you can transfer these embryos to the another female and allow that female to develop the embryo, because what is more important is that the embryo is of very high quality. So, you have generated the embryo in a good quality or hybrid female, and then ultimately you have transferred this embryo into a low profile females. So, in this process using an artificial insemination, superior breed male semen is used to fertilize the eggs in the female. Once the embryo is formed, it is transferred to another female which is actually going to be low breed, so that you do not have to spend lot of money on keeping these females to produce the offsprings. The donor female, so what is the procedure? The procedure is the donor female is treated with the variety of the hormones, so that it will produce several eggs, the process known as the superovulations. The egg is fertilized either by the natural intercourse, so that you can allow the males to go for the fertilizations, or you can go with the artificial inseminations. The fertilized eggs are recovered from the uterus of the donor animal using a catheter. The solution from the catheter is used to flush the uterus and fallopian tube. On average 6 to 8 embryos are collected in each flush. The quality of these embryos are going to be checked in the microscopy, and then the suitable embryos are going to be implanted into the recipient animal. It is important that both the donor as well as the recipient animal must have a synchronized estrous cycle for successful embryo transfer. So, if you see the procedure is very simple. First of all, what you are going to do is you are going to take the hybrid female, you are going to inject the some of the gonadotropin hormones, and once you inject the gonadotropin hormones, what will happen is it is going to start producing the eggs from the ovary, and then either you go for the natural in sexual intercourse, or you can use the artificial inseminations after the superovulations, and that artificial insemination is going to create the embryos. Now, what you have to do is in a non-surgical recovery of these embryos after the insemination. So, you can take after 6 to 8 days after the inseminations, you can recover these embryos, and you can use the catheter which is called as Foley's catheter to recover these embryos from the superior females, and then each embryo can be observed under the microscope for the classification of whether good quality embryos or the bad quality embryos. Then you can segregate the good or the bad quality embryos under the microscope, and these embryos can be stored in a liquid nitrogen either for the indefinite storage, or it can be stored in 37 degrees Celsius or room temperature for 1 day. After this, what you have to do is you have to transfer the embryo to the recipient surgically or non-surgically. So, you can do a surgery onto the recipient animal, and you can implant these embryos into the uterus, so that they will be going to develop. Then you can actually detect the pregnancy by palpation through the rectal wall after 1 to 3 months of the embryo transfer. In this step, you have to be ensure that the donor as well as the recipient females are going to be in a synchronous estrous cycle, so that the hormonal profiling of the donor animal as well as the acceptor animal are going to be same. Otherwise, in many cases what will happen is when you going to implant the embryos into the uterus, the female hormonal cycles are may not be in a conditions, or it may not be in a condition, so that they will not accept the embryos, and ultimately embryos are going to be removed from the uterus by the abortions. So, that to avoid the abortion like conditions, we have to keep the donor as well as the acceptor in a synchronous oestrus cycle, so that the hormonal profiling should match with each other. Now, after the a period of 9 months, the embryos are going to be developed into the calf, and by following these embryo transfer methods or embryo transfer technologies, you can be able to have multiple calves of high quality calves from the same hybrid animals. So, because you can actually have multiple recipients, and these multiple recipients are going to give you the multiple calves in a single year, whereas if you go with the natural process, the hybrider are going to give you only one calf in a single year. Now, let us go on to the role of biotechnology in the medical sciences. So, medicines are the class of molecule used to correct the disturbance in the host physiology. They can be chemical in nature to use to inhibit the aberrant enzymatic activity from the host, or they can be a chemical which going to be inhibit the activity in the pathogen. Both are these conditions are going to give you the normalcy in terms of the maintaining the inner homeostasis of the host. In few cases, host enzyme can be supplied as a drug formulations to drive the biochemical reactions. That is why the biotechnology has potentials in contributing into the development of the drug molecules. So, within the role of biotechnology in medical sciences, we are going to discuss about the 4 aspects. What we are going to discuss? We are going to discuss about the production of therapeutically important proteins. We are going to discuss about the gene therapy, vaccines, and lastly, we are going to discuss about the production of monoclonal antibodies and the role of biotechnology in these processes. The production of the therapeutically important proteins, a large number of genetic or metabolic diseases can be corrected by the supplying protein or factor. So, you know that the human body or in general even a single cell depends on the many of the metabolic reactions, and all these metabolic reactions requires enzyme. On the top, these metabolic reactions are controlled by the cell surface signalings, and these cell surface signalings are in turn being regulated by the different types of hormones which are going to be produced within the host. So, there is a complex processes in which the host is actually making a fine balance about the different types of enzymes which are available in driving the metabolic reactions, the amount of enzyme, the activity of the enzyme per se, and so on. So, all these enzymes are important, the hormones which are regulating these activities are very important, and the combination of these two are actually going to be used to maintain a normalcy in the host. What will happen is when you do not have any of these conditions, for example, because of genetic disease or because of the absence of an enzyme in a metabolic reactions, you are going to start developing the metabolic diseases or non-metabolic diseases. So, what will happen if you want to correct this? You have to use the biotechnology principles, and you can be able to provide these factors. So, what will happen is, suppose you are genetically modified or you are genetically mutant for a particular enzyme, or at a particular stage of your life cycle you are actually being programmed in such a way so that you will not going to produce that particular enzyme. In both of these conditions you are going to develop the metabolic diseases. So, what you are supposed to do is you are going to break that protein or factor you can produce in the bacterial expression system, and then you can supply these proteins or from that particular system in a artificial way, which means like you have to supplement these enzymes which are going to drive the metabolic reactions. So, in this particular type of approach, the gene of that particular enzyme or the protein factor is cloned into the appropriate plasmid to produce the recombinant clone, and these recombinant clones are going to give you these therapeutic proteins. And then what you can do is you can take this therapeutic protein as a supplement to overcome these defects, either it is genetic defects or the age dependent effects. For example, you might be producing the insulin when you are young, but in due course when the age crosses the 40 or 50, you have start producing lesser and lesser amount of insulin. In those cases, your body is requiring the insulin exogenously. So, that is why you are going to supply some amount of insulin, which you are going to produce in a recombinant system, and that is how you are going to overcome the deficiency which is being developed in your body. Now, let us take an example of the production of human insulin. So, insulin is a dimer of A chain and B chain which are linked by the disulfide bond. So, in a insulin you have two chain, one is called A chain, another one is called as B chain, and both are these A and B chain are being produced separately, and then they are being linked with each other by the disulfide linkages. You have the two disulfide linkages between the A and the B chain, and the molecular weight of an insulin is the 5808 Dalton, and the total length, total amino acid 51 amino acids are being present in the A chain and the B chain. So, what you can do actually is, if you want to produce the human insulin, you have to first clone the gene A, which is corresponding to this peptide chain. Then you have to clone the gene B in a separate clone, which is going to give you this peptide. And then what you do is you transform the gene A as well as gene B containing recombinant DNA into the bacteria, and then once you do that, you are actually going to induce the bacteria and you are going to produce the proteins. So, the bacteria is going to give you, and then you will purify. So, actually it is going to give you the peptide A, whereas the other one is going to give you the peptide B, and then what you do is you take the peptide A and peptide B, you recombine, and that is how you are going to have the insulin at the end. So, in a schematic representation, what you are going to do is in the insulin production is that in this process the gene A and the B is cloned into a bacterial plasmid separately to produce the two recombinant clones. Peptide chain A and peptide B is overexpressed in the E. coli and the recombined together to produce the functional insulin. So, this is just a simple example to say that how the insulin or human insulin can be produced in a recombinant expression system, and that is how you can be able to take this insulin to reduce the amount of insulin which your body is not being able to produce. We have many the list of the therapeutic proteins which are being produced and which are going to be, which are being in circulations to be used for overcoming the disease conditions is very exhaustive. This is a simple list with a classical examples. So, what you have is the factor 8 and factor 9. Both are these factors are required for the blood clotting. So, they are going to work in the case of hemophilia. Then you have the tissue plasminogen activator, which is actually also going to work in the case of thrombosis. Then you have the lactoferrin. Lactoferrin is going to work in the case of GI tract infections. Then you have the human protein C, which is also play crucial role in the case of thrombosis. So, and then we have the alpha I antitrypsin, which is actually is being used in the emphysema. Then you have the fibrinogen. Fibrinogen is actually required for the blood clotting. So, that you have to use for the wound healing. Then you have the Pro 542, which is actually required for the HIV infections. then you have the antithrombin 3 which is actually required for the thrombosis, then you have the collagen 1 which is actually required for the tissue repair, and then you have the serum albumin which is required for maintaining the constant volume of the blood. There are few examples I have taken, these are called recombinant chymosin. So, chymosin is required for manufacturing the cheese. A non-pathogenic E. coli strain coetal is used to use for the large scale productions. The recombinant enzyme is safe to use, cost less and available in abdomens. So, apart from the making a therapeutically important protein, you can also use or you can also produce the enzymes which are also very good in terms of economical usage. So, the recombinant chymosin is one of such example. Then we have the recombinant human growth hormone. So, recombinant human growth hormone or HGH, it is produced by the pituitary gland and hormone is required to support the growth and development of human. Recombinant HGH is cheap and safe to use for therapeutic applications. Then the third is the recombinant blood clotting factor number 8. In a normal individual, blood loss from a damaged blood vessel is prevented by the formation of a clot. The blood clotting is a series of reactions involving different factors. The factor 8 is deficient in bleeding disorders such as hemophilia, and recombinant factor 8 is going to supply to overcome the disease conditions. So, what will happen in hemophilia is that suppose you got the injury, you in a spontaneous process you are actually going to activate either the intrinsic pathway or the extrinsic pathway, and intrinsic or the extrinsic pathways are actually a series of cascades where you have multiple enzymes, and ultimately these enzymes are terminating to the activation of thrombin, and then once the thrombin is activated, it is actually cutting the fibrinogen, and that once you cut the fibrinogen you actually generate the fibrin, and these fibrin fibers are actually covering the clot or covering the wound, and that is how you are actually stopping the blood clot. But in these kind of disease conditions, one or other factor is missing, and as a result what will happen is that you are not going to be able to either complete these blood clotting cascades, or even if you will be able to complete, the reaction may not be that efficient. So, because of that you are going to lose lot of blood from the wound, and as a result the person is going to be it could be a life threatening conditions where the person may not be able to stop its blood clotting or stop the blood from the wound, and as a result you are going to lose lot of blood. Now, gene therapy. So, the production, whatever we have discussed so far is that you can produce and supply the recombinant protein, but that is a temporary solution for a disease which is actually genetically modified, which means if I am genetically prone that I am not going to produce the insulin, or if I am genetically if there is a genetic defect that I cannot be able to produce the factor 8, then even if I supply the factor 8, it is going to be a temporary situation, because you do not know when you are going to get the injury, when you do not know when you are going to have the meal, and also all things. So, and then on the other hand you might have to carry these drugs along with you all the time, while because if there is a requirement, you have to take these medicines, or you might have to take these proteinaceous factors to execute the processes. Whereas, in the gene therapy approach, what you can do is you can actually take the gene and start expressing the protein in your body itself, and that is what the procedure is called as the gene therapy. So, these solutions are temporary for the treatment of a disease conditions. So, alternative is that the human expression system can be used to produce the proteinaceous factor after inserting the recombinant clone into the human cells or inside the human body. Recombinant DNA is packed into the appropriate DNA delivery system, either a virus or a liposome mediated system, to deliver the gene into the human cell to correct the mutated gene, or encode a therapeutic protein drugs to provide the treatment. When you talk about the delivery, it is also important that you do a delivery to a specific cell. For example, if we are talking about the insulin, we have to deliver the cells, we have to deliver these recombinant clone to the beta cells, because beta cells are the cells which are actually being trained to produce the insulin. We cannot produce the insulin in muscles, or you cannot produce the insulin in brain, because that is not the purpose of these cells, because if you do so, you are actually going to also interfere the metabolic reactions which are occurring in these cells, and as well as you are going to disturb the physiology of that particular organ. So, that is also very important, that you actually not only going to transfect the cells with the help of viruses or the liposome, you also should do the targeted transformations or targeted delivery of these recombinant clones. Gene therapy is of two types. Gene therapy could be of somatic gene therapy. So, somatic gene therapy is that in this therapeutic approach, the therapeutic genes is transferred into the somatic cells as per the requirement of the individual to treat the functional defects. This treatment does not move to the patient's offspring or next generation. For example, so the example of the somatic gene therapy is that the diabetes. So, if somebody is suffering from diabetes because it cannot produce the insulin, then what you can do is you can recombinantly clone the insulin A and B gene into a recombinant DNA, and then you deliver that to the beta cells. But the so the advantage is that that particular individual is going to start producing the insulin, but in this particular type of defects are not going to move to the next generation, because the transfection as well as the integration of this particular gene into the genome which you have planned is going to happen only into the somatic cells. It is not going to happen into the germline cells, which means this information is not present in the germline cells. So, it will not transfer from one generation to another generation. So, it will going to give you the permanent cure for that particular individual, but it will not go to the as a cure to the offsprings. Whereas, in the germline gene therapy, in this therapeutic approach, the germ cells, for example, sperm or the egg cells are transformed by the introduction of the required gene to produce the protein or correct the mutated gene. So, in some cases people are only having the mutations, they do not have a problem of overexpression or expression of this particular gene, they have a problem of that they have a mutated gene. So, in those cases you can actually make the correct gene, and then you can use the homologous recombinations to replace this gene into the genome, and that if you do that procedure into the cells of the germline, for example, if you do that procedure in sperm or the ovum, then it is once the embryo is going to form, this information is going to replicate into the all the cells of the offspring. So, any kind of gene therapy when you do in the germline, it is going to propagate for the many generations. This allows the treatment move to the patient's offspring or to the next generation. Although the approach seems promising in providing the long term solution to treat genetic disorder, but there are several ethical, technical reasons and possible future risks. So, it looked very promising that you can use the gene therapy, you can replace or you can do whatever kind of genetic manipulation of the human beings, but it has lot of ethical issues, it has lot of social issues, and at the end it also has many of the technical obstacles to overcome, and that is how this that's how the gene therapy also has many disadvantages. So, the technical problems associated with gene therapy is as follows. Number 1, it is short lived. The therapeutic genes delivery into the cells give the short term effects either by rejection of the recombinant DNA or suppression of the gene expression. Due to this problem, patients need to go for several rounds of gene therapy. So, what will happen is, suppose you have given one round of injection to the patient and then the virus has gone to the beta cells, it has started producing the insulin, but in due course what will happen is, because these are recombinant DNA, the system does not may not actually accept these exogenous DNA. These cells either these cells or the DNA itself is going to be short lived, and because of that the in due course the effect is going to be over. So, because of that you might have to need to have the multiple injections to maintain the level of protein productions. The second, which is actually more serious, is the immune reactions. The virus containing gene is treated as the foreign object and the immune system is stimulated to attack the invader. It is the main reason of the reduced effectiveness of the gene therapy. So, this is actually a very serious problem, because once you inject the virus containing genes into a human being, the human body has a very pronounced or very good reliable immune system. So, these immune systems are going to recognize the viruses as the foreign agent, and because of that they will actually going to either remove these viruses from the circulations, or they will not allow these injected viruses to propagate and provide any kind of the long term effects. Then the third, the viral vectors. The viral vector used to deliver the gene causes much adverse immune reactions and toxicity in patients. So, some of the people are very sensitive for virus as well as the viral derived products, and those people are going to show severe anaphylactic shocks, and sometime it may actually lead to the death of that particular individual as well. Number 4, the disturbance of the host physiology, because whatever you are doing is actually a exogenous thing, it is actually going to disturb the human physiology. So, if the gene integrate to a wrong place in the genome, it may cause functional defects. In few cases, it may disturb the function of the tumor suppression genes, results into the development of tumor. For example, X linked severe linked immune deficiency (XID) patients injected with the transformed hematopoietic stem cell led to the development of the T cell leukemia. So, there are two things could happen. One is that because you are actually putting an extra burden of the viruses, extra burden of the recombinant DNA into the system, and as well as you are actually disturbing the metabolism by providing a exogenously supplied enzyme, you are actually going to disturb the physiology of that particular individual. And you know that the protein production from your recombinant DNA may not be having the same tight control compared to the naturally occurring the gene. So, because of that also you might be producing very large quantity of that particular protein, or you might be producing very small, or you may not be producing as per the requirements. For example, if I need an hexokinase at time 0, or if I need an hexokinase when I have taken a breakfast, but the hexokinase level might be very high even from the throughout the day. So, that actually is going to disturb the overall metabolism of that particular cell as well as the as an individual in totality. The other aspect is, suppose we put the homologous sequences to these genes and ask them to go for recombinations, and if there will be any mistakes in this recombination process, they may go and integrate into the genome and disturb the existing genes, and because of that sometime what happen is they might be actually disturbing the tumor suppressor gene. So, if you actually going to remove the tumor suppressor gene from the system, you are spontaneously going to evolve, or you are spontaneously going to develop the large quantity of tumors, and that is what exactly happens in the XID patients. If you take these XID patients and if you inject them with the hemopoietic stem cells, they actually eventually develop the T cell leukemia. So, because of these kind of technical issues and because of these kind of problems, so the gene therapy does not have any futures in terms of providing the permanent solutions for the mutations or the deficiency in having the short supply of that particular recombinant factors. Now, let us move on to the vaccine. So, vaccine is given to the develop immunity against the disease in the human or other vertebrate animals. Vaccines are of different types. Vaccines are dead, attenuated organisms or protein derived from them. There are different strategies to enhance the immunological responses to give long lasting protection against the disease with minimal adverse effects. So, vaccine are the molecule which are actually going to train your immune system. So, they are going to train your immune system so that your immune system is going to work against the particular disease. So, right now the disease causing agent may not be available or may not be present in the host or present in the human being, but once the with the help of the vaccine you are actually training your immune system to combat the infectious disease caused by any kind of agent. Then once this infectious disease agent comes, it actually going to destroy this agent spontaneously. So, the purpose of a vaccine is that it will going to give you the long term immune response, and that immune response is going to give you some kind of protection against this particular disease causing agents. And in this case you can actually take the dead organisms, you can take the attenuated organisms, or you can even take the proteins from that particular organism to challenge your immune system, or you trained your immune system in such a way that when the real organism is going to come, it is actually going to combat against these infectious organisms, and that is how they are going to eradicate these infectious organisms from the system. So, there are different types of vaccines which people have developed for vaccinations. You have the killed vaccine. So, in this vaccine preparation, the pathogenic organism is killed by the chemical or the UV treatment and used as an immunogen. So, once you used these killed organisms as an immunogen, it is actually going to increase your immunity against this particular infectious organism. This killed organism is mixed with the adjuvant to enhance the immunological responses and the long memory, which means if you mix the killed organism with a adjuvant, you are actually not only going to enhance the immunological responses, you are also going to generate lot of memory B cells, and because of that you are going to have the very long memory to combat against this particular infectious organism. The second is you have the attenuated organisms. In this vaccine preparation, organism is treated with a chemical to destroy its ability to cause disease. One of the classical example is the polio virus. So, the polio vaccine, the polio drops what we take in for the polio vaccination is nothing but the attenuated virus, which this attenuated virus means it will still be able to propagate, but it will not be able to cause the disease. As a result, organisms grow and give stimulation to the immune system for a long term immunological memory. So, once you give the polio drops to the baby, it is actually allowing this particular virus to replicate into its gut. It allows this particular virus to remain in the system for very long time, but because this virus is attenuated, it cannot cause the disease. The baby is keep generating the antibodies, keep generating the other kind of immune responses against these viruses, and as a result at the end it is actually causing the very robust immune response, it actually causes a large production of memory B cells, and that is how the baby is going to have very long term protection against the disease. Then you have the toxoid. In this vaccine preparation, inactivated toxic compound are used as an immunogen to train the immune system. Then you have the subunit vaccine. In this vaccine preparation, a pure protein or antigen is given as an immunogen. It is the safest form of vaccine with minimal side effects. One of the classical example of subunit vaccine is the hepatitis vaccine. The vaccine what you use for hepatitis is actually the hepatitis surface antigen what you have cloned during the using the recombinant DNA technology, and then you have produced the protein like the surface antigen, and these surface antigen you are injecting into the human beings, so that the body is developing the antibodies against that surface antigen, and that is how you are actually developing a robust immune response against the hepatitis virus. Then you have the conjugate vaccine. In this vaccine preparation, the bacterial coat is tagged with the immunogenic protein to induce production of immune response against the bacterial coats. If you would like to be interested to know more about the vaccine, these different types of vaccines or how the vaccination is being done, you can follow any immunology textbook, such as you can follow the Reut's immunology, or you can use the Janis Cuby's immunology, and any standard immunology book can be refer for this purpose if you would like to be interested to study more about the vaccinations. Before we go into the monoclonal antibody productions, what we have discussed so far, we have discussed about the applications of the biotechnology in the medical sciences. We have discussed about how the biotechnology has allowed the production of the different types of the vaccines, how it has allowed the production of different types of therapeutically relevant recombinant proteins, and how the we are with the help of the biotechnology we are actually performing the gene therapy applications. So, with this I would like to conclude our lecture here. In our subsequent lecture, we are going to discuss about the production of monoclonal antibodies, as well as the some more applications of biotechnology in the other field as well. So, with this we would like to conclude our lecture here. Thank you.